Hi guys, here is the notes for DNA structure and replication. So this video will talk about the shape, what DNA is made up of, and a brief introduction to how it copies itself. So let's start out with a definition of DNA. DNA stands for deoxyribonucleic acid, DNA. Now, it is the genetic information that carries information about an organism from one generation to the next. So that is what DNA is. It carries information about an organism on to the offspring, to the child. Now, DNA is found in the form of chromatin in the nucleus of a cell. So DNA is found in the form of chromatin, which is a loose, unbound strand that looks kind of like spaghetti. And they are found in the nucleus of the cell. So that's what it looks like from an outside perspective. If we were to use a microscope, we could see the DNA inside the nucleus if it was a high enough power microscope, and it would look like a big jumble, like a big mess in there, kind of like a bowl of spaghetti. Okay, if we were to look at the individual structure of one strand, it looks kind of like a twisted ladder. So a DNA molecule looks like a twisted ladder, which is made up of three things that make up a nucleotide, which is made up of units called nucleotides. That's the individual piece. Okay, that individual piece is made up of different things, 3 different things in fact. Each nucleotide is made of 3 components. They are a 5-carbon sugar molecule, Five carbon simply means that the molecule has 5 carbon atoms. A 5-carbon sugar called deoxyribose. It's made up of a phosphate molecule, and a nitrogen base. which makes up the rungs of the ladder. Now there are 4 different types of nitrogen bases. Four different types. They are adenine, thymine, guanine, oh, that didn't show it very well. I'll go over that with black. Guanine and cytosine. So those 4 nitrogen bases can be found in DNA molecules. They are the rungs of the ladder, which we're going to draw that on the next page. So we're going to draw the individual nucleotides up here. All right, then we're going to talk about, down at the bottom, how the DNA actually replicates. So we're going to start with our phosphate molecule, which is a circle. Okay, that's phosphate. I'm going to represent it with the letter P. The phosphate is bonded to our sugar molecule, our deoxyribose, and it is a pentagon that looks like this. So this is deoxy. Okay, the deoxyribose is then bonded to a nitrogen base. I'm going to start with adenine. Okay, and it kind of comes out like this. And the adenine, well, actually it has a little bond area so that it will only bond with thymine. Okay, I'm going to use purple to represent my thymine. Alright, and it fits together in such a way that it can only bond with adenine. Okay, now the structure of DNA is so that there are 2 sides and they're running parallel to each other, but they're facing opposite ways. So it is said to be anti-parallel. Okay, I'm not going to test you on that. It's just a fun little side note. So if you're wondering why I'm drawing my nucleotide going the other way, that is why. Okay, so here we have our other deoxyribose that is bonded to another phosphate over here. Okay, so we have 1 nucleotide here. So this is 1 nucleotide. This is also 1 nucleotide. All right, and they are bonded together to other ones going this way, to other nucleotides. So we're going to draw another one of those. Okay, so I've got a phosphate that's bonded to this deoxyribose. It's bonded to another deoxyribose. And let's say that we have cytosine this time. Okay, I'm going to draw my cytosine in yellow. That is not showing up very well, so I'll switch to black. So cytosine, right, it has a different shape from the adenine and the thymine, so that it can only bond with the guanine. So I'm going to make it kind of pointy. It's going to bond with my guanine, which is going to be I already used purple and green, so let's use orange. Okay, guanine is going to be orange. And notice that it fits with cytosine. So guanine is then bonded to a deoxyribose. Alright, this pentagon is not going to be great, guys. Sorry about that. Alright, and it's bonded to this phosphate here and to another phosphate. Okay, once again, each one of these is a single nucleotide. So here we have a nucleotide, and here we have another nucleotide. So we have the 4 possible nucleotides represented here. Now, let's dive right into how DNA can replicate itself. So during DNA replication, a DNA molecule unzips like a zipper on a jacket. It unzips between its nitrogen bases new bases or nucleotides pair up with the old bases, pair up with the bases on the old strand of DNA, the old strand of DNA, forming 2 identical DNA strands. Alright, I'm going to attempt to draw down here what it's going to look like. Alright, yours does not have to look exactly like mine. If you're having trouble drawing it, I can help you do it in class tomorrow. So there's one side. Oh, this looks terrible, guys. Sorry about that. And here is where it's unzipping. Okay, so we have new strands of DNA forming down here. All right, and between them, they have nitrogen bases like adenine, thymine, cytosine, and guanine that look like rungs of a ladder. So that it looks like this. Okay. Now, I think I can do a little bit of a better job on that part. So let's see. I'll just draw it down at the bottom of the page of the undivided part. There we go. That's much better. Let's see if I can do just the whole thing. Oh, that's a much better one. All right, you guys can just cross that one out. Okay, so it will end up looking something like this. So here is our old strand of DNA that has not yet unzipped. It will, don't worry. And here we have our new strand, here and here. These are going to be exactly identical. There's not going to be any difference at all between this strand and this strand. So this is how the DNA is replicated during the S phase of interphase during the cell cycle. If you guys have any questions, let me know.